Hey, everyone. Erez, what's your team number? Oh, 11?
Yeah, I don't even know. 11 maybe.
It's 11. So we have received documents from teams 1, 2, 3, 5, 6, 8, 10, 11. We're missing 4. Sonia, Lily, and Jonathan are. Oh, Lily, did you guys upload your end-to-end pipeline?
We were not able to do it because when we met with our project mentors to go over what we had so far,
Yeah.
they told us that we wouldn't be able to do the project with our current data because our project was to do like a diagnostic model while the person was alive, and the data we had was from gray matter from postmortem brains. So it wasn't going to work. So we're in the process of finding new data. They gave us a lot of avenues to explore, and
Good. Who's your mentor?
Carle and Neil.
Okay. I'm surprised. So what data don't you have?
Epigenomic data that's from either cerebrospinal fluid, blood, or plasma, and then the corresponding transcriptomic data.
I see. So basically what you're looking for is not only pathology, but and transcription, which we have both, but also some kind of biomarker.
Yeah.
I see. So Lei Hao in the group has H3K27 acetylation epigenomic data from blood, but we don't have postmortem brains for those.
Oh, we don't want postmortem brains. We would like to look at, because there is data with epigenomic data that corresponds with Alzheimer's disease from blood or CSF or plasma.
Okay.
And we can't, and we found it for like just epigenomic data or just transcriptomic data, but we haven't found one that has both from the same people.
Okay, hold on one second. So this is, I don't even know what round we're at. So, oh, maybe it's 5 or 6. It's 6. So it's end to end Python. Okay, so team 4, right? No, you're not team 4. You're team. Gosh,
No, we are
yeah,
team 4.
you are. Sonia and Jonathan. Okay, so talk with Lei Kou. He has AD status and H3 K27 isolation in blood. This might be helpful.
Yeah, that'd be so helpful. Thank you.
And then these are just some. I'm sending you a link to this document. You can share it with the rest of your team, but you know, keep it confidential, of course. I sent you 2 links to various presentations on this. It can help you sort of wrap your head around the data set and stuff.
Yeah, that'd be great because it is a little difficult to interpret when they don't provide like explanations for what the abbreviations are.
Yeah, yeah. Okay. But reach out to Lei. I think his email is leihu. Is that right?
Yeah, that's perfect. Thank you so much.
And that is why we're doing this end-to-end pipeline. Because I don't want it to be week 14 and then you're like, turns out we don't have the right data. Okay, cool. So then team 7, that would be Evelyn, Reina, and Jason. Is either of you there? Do we have a TA on? Sorry, guys. If one of our TAs were there, it would be much easier. Okay, so thanks Fatima for the upload. If anybody hasn't uploaded and you're there, please upload now. ¿Me llamo? Nada. Nada. Wow. All right, so let's go through the folks who are here. So team 1, Jenny, Jojo, and Shin, are you guys here? Is any of you here?
Hi. Yeah,
Awesome.
me and Jenny are here.
Perfect. Okay, so let's see. We feel interested for factor binding profiles and functional categories, regulatory elements, AML, UML. Here's the link to the slides. Beautiful. I totally love it. How are you guys doing on data?
Yeah, so we can load the data and we can run through the deep learning pipeline.
Beautiful.
But yeah, it just seems like because we also plot confusion matrix, it seems like it's not predicting very well for now.
I mean, that's fine. Basically, that's the whole point of the project. So if it was predicting already, you'd be like, well, that's a little too easy. I mean, biology is complicated. You might not have all the features. My guess is that as you start gaining more insights as to why is it not predicting and is anything correlated with output, etc., then you'll have better ideas how to do feature selection, etc. So, I would keep pounding at it, but I think the most important thing is that you have all of the things in place, basically. Um, Beautiful.
The bulk ataxic peak value, how one
I love that. That is so cute.
So the first validation and test, and as you can see here, panels because they represent ATGMC.
This is awesome. You guys are in such good shape. I love it. Any questions for me, or you're all set?
I think we have a small question in terms of mentoring. So we've showed some emails to folks in your lab, but we haven't got any response.
Who did you email?
Jojo, who did we email?
Sorry, I can't remember them. I think Sean, and also, yeah, sorry, I forgot the name.
I think Sean may be gone, but I think he may have graduated and left. So he was around at the beginning of September, but he may have just not checked his email lately. So, but are you thinking about Shushen?
Oh, the other person is Carlos.
Carlos, you should be able to hear back from Carlos. He's usually responsive. So I mean, email them back and CC me. But I think so. Shushen's wife is in labor, so he may not be available. And then they had a big presentation for next week at American Society of Human Genetics, so they may have been busy for that. But Carlos is usually very responsive, and I mean, Shushen is amazingly responsive, so you should be able to hear back from them.
Okay.
Okay.
We'll try.
All right. So team 2, Jessica, Ayush, and Mindran, are you guys there?
Yep.
Awesome.
Yeah, we're here.
Πού είναι πάνω? Η μαμά είναι εδώ. Πού είναι η μαμά? Ναι, πες της ότι είμαι στο μάθημα και διδάσκω. Εντάξει? Beautiful. Oh, lovely. Nice. Nice. So you've been able to load up all the stuff and get some predictions going and all of that, right? Ayush? Oh, I'm sorry. I was clicking unmute as you were clicking mute, so I muted you again. I'm sorry.
Okay. Yeah. So the Colab notebook runs. There's like some small issues with some files not getting imported properly, but we expect to run pretty soon.
Awesome. You guys are in such good shape. This is awesome. Good. Any questions, or you're all set?
I think we're mostly good.
Okay. Awesome. So team 3, Landon, Ari, Maya, are you guys there?
Yep, I'm here.
Awesome. So can you turn on your video so I can see your face?
Yeah, one sec. I just had to plug in my laptop.
Of course, of course. There are upsides to Zoom, but the downside is sometimes I don't see. Cool. So role of selection driving the convergent evolution of monogamy. Oh, this is awesome. Good. Ναι. Η μαμά δεν είναι εδώ. Δεν μπορείς να της πεις, βρε αγάπη μου, έχω ζοκίτα, τόσους σπουδαστές. Έχω το μάθημα τώρα. So this is only based on sequence or expression, or what is this based on?
It's just based on the sequence, but the data set comes from like transcriptomic data. Yeah, so it's just looking at CDSs.
But why not just look at expression at the same time?
That other people have done that for like in a limited way. I don't actually think we have the expression level data is the issue.
Hold on one second. Okay. Sorry guys, I'll be back.
Did that just really happen?
Sorry, my wife is presenting somewhere, so she's a little stressed and kids needed something, so it's all set now.
No worries. The data doesn't have, I don't think all of the transcriptomes have expression levels. So it's not like necessarily quantitative.
Okay. So basically then the idea would be simply positive selection or something?
Yeah. Looking for genes under positive or negative selection and then comparing.
Yeah, in both species, basically. Very cool. And let's see. Do you have the data? Is it running?
We don't have, we're waiting for one transcriptome from someone in Lambda and like someone Lambda and Maya's lab collaborates with. A lot of the end-to-end pipeline focus was on the like dealing with the fact that one of the genomes wasn't annotated. Yeah, this will explain.
Yeah, I would love to see some code running and stuff like that. Do you have?
Yeah, no, there is code running.
Okay, but it's not in the video.
We have a schematic, and you can look at the GitHub. There should be a screenshot of the GitHub.
Okay, sounds good. But you've been able to sort of find all the right data, load it up. You have code.
Yeah.
Putting that together.
Yeah, the GitHub is linked in the document.
Okay. Again, this is all to help you guys. You don't have to convince me. You just have to convince yourselves. Are there any sort of points of failure that you can foresee now, or you think the data will come in place?
We did have to deal with the annotation, the issue, trying to deal because one of the genomes we're working with wasn't annotated. So there was like a difficult method trying to like do something that would compensate for the fact that it's not annotated, but I think we're in good shape.
Okay, sounds good. Manu, I just responded to you. I hope that's okay.
Yeah, I uploaded the link. Thank you.
Okay, perfect. Thank you. All right, so then you're all set, right?
Yeah.
Perfect. Good. So yeah, just make sure that you have everything in place. I don't want any surprises at the end where you're like, oh gosh, we can get access to this or that.
Yep.
Okay, so is anybody here from Team 4? Sonia, Lily, and Jonathan, I guess you guys did not upload something yet, right? Yeah.
Yeah, we really couldn't. I mean, we could write out like the strategy we're gonna do,
no. My goal is not for you to write out something to make me happy. My goal is for you to make sure that everything's in place. So once everything is in place, then write something. If everything's not in place, that's exactly the goal of this exercise, to identify what's not and then to fix it. So I hope that it works out with Lei, and if not, we'll figure out alternative strategies. So Kathleen, Alice, and Stephanie, are you guys there?
We're all here.
Awesome.
Yes.
Do you have the video up there as well?
We only show the result of the code though, rather than showing when the code runs.
But you have been able to load up what you needed?
So we have some problems. We're not able to get access to the whole genome sequencing VCF data from GTEx because they're all in dbGaP.
Yeah. So my team should have access, and I think Yongjin would be the right person in the team, but he just started his faculty job. I'm trying to think if anybody else has access. So let me just, check real quick, and then. So what do you need from GTEx?
The whole genome sequencing VCF files to get the allele frequencies.
Okay, let me get Austin and okay. Sorry, I have way too many windows open because of Zoom. So, okay, there you go. Is it okay if I include the link to your project proposal, etc., so that they know what it's about?
Sure.
So if you don't mind responding to that email and sort of providing a little bit more info, because mine is a little curt, you could just say, hey, we're interested in doing this. Here's the papers we've looked at before. And you've seen the rare variance effects paper by Stephen Montgomery and others, right? In GTEx.
We have not.
Okay.
Oh wait, is that the? Oh, that's the one. That's the paper that we've been using.
Okay, good. So you've seen that one.
Yes, we have.
Okay, perfect. Okay.
Also, one thing, we might need to get data from UK 10K, and it says that we need to
UK Biobank or UK 10K?
Wait a second. It says from this website on the chat. So we have reached out, and then they mentioned that we should have like a nominated institutional representative to sign agreement.
Yeah. I mean, we have such a thing, and we could add you to our team. So, you know, I think it'll be much quicker if you go through Remy.
Thank
Okay.
you.
All right. So I hope this helps.
Thank you very much.
Thank you so much.
Of course. Okay. So
Thank you.
6, Joseph, Kyriakos, and Pablo.
I'm here.
Hey. So that's awesome. So you have data?
Yeah, we have data that we can load in a standardized way. And we still have a few more data sets that we want to download. And it's for like, the purpose of that is to like push our model to the limits. So we're still acquiring those, and all of them are on GEO.
Did you upload your video or not yet?
Oh, we did.
You did?
Yeah, it's the one with Pablo Villalobos. Yep, that one.
Ah, perfect. Thank you. It's Team 6, right?
Yep. So we have a basic version running. Yeah.
Nice. Beautiful. Nice. This is awesome. Good. And I mean, I find this just so helpful. So thank you guys for putting this together. Any questions, or is everything okay?
No, so far so good. Yeah, we're just one, I think, problem or challenge, I guess.
Yeah.
Is that the batch correction methods that we want to input our lower dimension to uses Euclidean distance usually, but our learned latent space, there really isn't a concept of Euclidean distance. So we might have to.
No, but you can define your own distance metric. It doesn't have to be some distance.
Right, yeah.
Thing. You can just, arbitrarily define any distance metric in any set of points, right?
Yeah, so that's one thing that we're going to have to look into.
That's one.
Other than that, it's going as planned so far.
That is awesome. This is great. So Team 7, is anybody from Evelyn, Raina, or Jason here?
Yeah, we're here, but we haven't put our document for this weekend here, but we are putting them into a PDF right now.
Okay, you're going to upload a PDF before the end of the session?
Maybe. Okay, so we can soon share what we have right now, but we haven't compiled them into a PDF yet.
Yeah, if you compile it into a PDF, we'll come back to you.
Okay.
Okay. And so basically, let me go through the other teams, and if you manage to upload a document, it will just appear on my file right there.
Okay, I see. Got it. Thank you.
Awesome. So Team 9, use row 13. Okay. Team 10, Thomas, Wendy, and Hugh.
Yep. Hello.
Hi. Nice. That's great. And you have data? Is the code running?
Yeah, the data, we have the data. Actually, Hugh basically did all the data work, which is great. And in our video, we just kind of run example subsets of the code so that
it
doesn't take forever, but yeah.
Okay, my video player is having trouble. Yeah, I think the video format is not working very well for my player. Anyway, it's
Hello, everyone.
yeah, there's something wrong with the video. I'm not able to scroll forward, and I'm afraid I can't spend 5 minutes just waiting for it to load. But tell me about your data. So you know, are you able to sort of load it up, run it, get some initial pictures that things are working?
Yeah, we use the data from the paper, and like they already pre-processed the data. Yeah. So like I managed to run the data.
Okay, good.
Yeah.
So it runs in your hands, and you can sort of generate some kind of visualization of the data that, you know, you have what you need, basically.
Yes, but I feel like we are like having like not enough.
You have too few data points. Is that it?
Yes. So if we want to have more data, we have to like do all the pre-processing again from Geo.
Oh, really? No,
Yeah,
I did not go there. So the paper that you're using, you're saying that that paper did just not have enough individuals.
Actually, like they have another, like what they call is like raw data. They already pre-processed some of them, but like they do not label it clearly, so that it's very hard to know.
I see.
What is the meaning of the columns and the rows?
I mean, one approach is to just do it yourself. Another approach is to look for another paper.
Okay.
I'm very tempted to sort of go with the latter, because there might be a reason why they didn't process everything. Like, it just might not be easy. Do you see what I'm getting at? You can also contact the authors and just say, you know, hey, we weren't able to see this. Does that make sense?
Yeah.
That makes sense.
Thank you.
Sure. Okay, very cool. Yeah, let's see how it goes. But you know, right now you seem to sort of have a path for overcoming the challenge. So I would love to know that you've managed to overcome it. So is that all right? Any other questions, or you're all set?
I think that's mostly it.
Okay, sounds great. And here, are you in China right now or something?
No, I'm in Vietnam.
Vietnam, but it looks like the middle of the night. So thank you so much for calling in despite the crazy hour.
Yeah.
He carries our team.
Thanks, Jerry. That's awesome. All right, so team 11. Let's see.
Yes.
Nice, 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 nice. Very cool. So you guys seem to be in very good shape. Do you have what you need?
You asked me what I need?
Yeah. Do you have what you need in terms of data, in terms of code, in terms of, you know, pipeline?
I think so. I think we need to find some computational resources, because the files, you know, it came out to be 72 gigabytes after processing. So it's just a lot of data to play around with, surprisingly.
Okay.
So we need to figure out where we're going to run stuff and so on. But
do they have it anywhere in any cloud?
Pardon me? Do you have that where?
Do they have it in any cloud?
No, I think it's right now like sitting between our computers, but the original data in the script is on a Dropbox. So we can just run it from wherever.
Yeah. Okay. Is there anything I can help with, or you feel that you have a good handle on everything?
I think we have a pretty good handle, nothing specific. I know that some of my teammates who are in Europe right now have challenges showing up for this because it's very late on their Friday.
Yeah.
But other than that, we just, we need to work through the data. I think it's not going to be easy. Probably the next thing we're trying to find is a database that kind of now connects to these kind of genetic fragments into genes and motifs and so on, and how they're all related.
Yeah. And do you have a mentor that you're working with?
Yeah, like mentioned there, that we have both Nason and Shushan.
Yeah.
I think I it auto-corrected wrongly his name, but they've been very helpful in finding the information and everything else. So
great.
I'm thankful for their time.
Yeah, good. I'm glad. And now I see 13, which says Joe, and then Joe is the got it.
Yeah, so we changed that also, but we uploaded the.
There's a lot of background noise. Okay. So you guys are now doing the drug repurposing?
Yes.
Nice. All right, let's see what you got. So this is team 12. Did you guys upload your video?
We didn't do the video, but we uploaded a document with links to our notebook.
Okay, that sounds great. So let me go to that.
It's the first one, the Manu. Yeah.
Perfect. Yes, you messaged me about that.
Yeah.
Yeah, I'm sorry, it's silly, but it does help. That's what you meant. Yep. Nice. All of this is code that you guys have written now?
Well, we used parts of it. So there was one graph neural network paper that predicts drug side effects. So we kind of repurposed that one.
Very nice. And do you have data from the drugs and all of that?
Yeah, so the SNAP library on which people built this like polypharmacy side effects things already has a lot of PPI information.
Nice.
So we use that data. I think we would like to kind of test it on other data sets that are not in Decagon. One interesting data set would be the SARS-CoV proteome map, where they use that to do drug repurposing for SARS-CoV.
I love it.
Yeah.
It sounds fantastic. Good. So you're in good shape, right? Do you need any help or anything like that?
I don't think so. Thank you.
Okay, awesome. Good. And then Manvita, Kamal, and Joe. Oh, wait.
That's us.
That was you, sorry. Guillaume, Na, Juna, and Fatima.
We're here.
Okay, good. And then you've uploaded your stuff there. Nice. Oh, this is lovely. Do you have a video uploaded? Oh, there.
Yes.
Very cool. So you have the data you need, you have the code in place, you've already loaded it up, and you have all the components?
Yes. Yeah.
Nice, nice, nice. So you guys are in good shape. Any questions you have or anything I can help with, or you seem to be rolling?
I think we're good.
Awesome. Good, good going. And then 15, Kamal, you're now part of team 13. Are these the same links? Kamal, are you there?
He's not here. Yeah, sorry.
But he's part of your team now, or?
Yeah.
Yeah.
I see, I see. So I can just delete this, or?
Yeah, you can delete it.
Okay. And then I'll delete this one, and you guys from now on will be team 13. Is that okay?
Yep.
Awesome. Wow. So are there any teams who haven't gone or folks who haven't spoken? It seems like everybody has. So Tatiana, did you reach out to the folks who are looking for team?
No, I didn't. They still didn't respond me. Can you tell me their emails? I can't find them. I have written to them in chat and I don't know. It seems like both of them are not actually here because neither Stuti nor Andrew didn't respond me.
We are here. We just need a second to think because it's just happened.
Okay, sounds good. So yeah, you guys, you know, it'd be great if you could figure out a way to work together.
Can you send me the emails so that we can maybe write each other something regarding this?
Have you emailed them already, or have the TAs emailed them already?
I think the TAs have emailed them. I haven't. I have just written them in chat right now.
Okay. So again, I don't want to put them on the spot, but you know, I'm sure they'll get back to you, or you know, I think what I would encourage you guys to do is look at Tatiana's profile and background, etc. I'm sure she will be able to do something great for your team. Oh, I see. So there you go. I just got your text. Christian from team 12. Stuti and Andrew. Okay. So yes, Stuti and Andrew, we can chat after this if you want, and then figure out a path. Actually, you know what? Let me create a breakout room. So I think that so Tatiana, stick around for a little bit, and then Stuti and Andrew, please stick around for just a little bit, and we'll talk about that just as a sort of side conversation. But and then Kathleen, I see that you put the EBI link there. I guess we've already talked about that, right?
Yes, we have.
Yeah. Okay. Perfect. Guys, you're now off on your own. Can everybody turn on their video? I'd love to sort of see your faces. So basically, we've basically reached a major milestone where there are no more milestones. So the next thing is going to be at the end of the tunnel. So basically, the next one is in 3 weeks. So basically, not next week, not the week after, but in 3 weeks, and it's going to be the mid-course report. And basically, what I'm describing for a mid-course report is that between now and then, and you have 3 weeks to do so, I would like you to sort of somehow make enough progress that you can see the end of the tunnel, to make enough progress that you can basically say, this is what the figures will look like. This is what the main findings will be. So that when you get to that point, there's basically no more new science, that it's more wrapping up the stuff that you've already gotten to. So basically, the next 3 weeks are all exploratory analysis. I expect you guys to sort of continue meeting regularly. I hope that you are now used to that, because we've had milestones every week. So I hope it kind of forced you to sort of really become a well-knit team. But don't drop your guard now. What I want you to do is somehow keep pushing with your own milestones. Basically, the next milestones are the milestones that you guys defined in your own project. So use those as internal deadlines to continue making progress. And that's part of the learning experience. There's a learning experience of having a professor meet with you every week. And there's another part of the learning experience, which is the self-motivation, let's meet our own deadlines part. So do this for the next 2-3 weeks and try to get at a point where you can kind of see, here's what our report will actually look like. And then when you're there, the importance of having that mid-course report milestone is that very often students basically spend 90% of their time working on aim 1, and then 10% of the time working on aims 2, 3, 4, and 5. Sorry to be so blunt, but kind of like me during lecture, where sections 2, 3, 4, and 5 are covered in the last 15 minutes, and the section 1 takes, you know, the 4 hour and 15 minutes. Anyway, it's not that bad. But I don't want you to sort of cram everything at the last minute. I want you to sort of really space things out and sort of realize that, wait a minute, aims 3 and 4 require a massive amount of work, and we've only worked on aims 1 and 2. So basically, I'm hoping that by the mid-course report, you guys are at the point of having an outline and having a detailed sort of bullet point list of all of the paragraphs and what they're going to look like for your final report, and having most of the figures ready, if not all of the figures ready, or at least sketched out, so that for the last 3 weeks of the course, or 2.5 weeks, you're able to just wrap, basically figure out what remains to be done and just finish that. Is everybody clear as to sort of what the next goals are ahead? Yes, no? Good, awesome, very cool. Lovely, so we have 88, 13, 000. Any questions? If not, everybody except the 3 people I mentioned, you guys are all set. So see you in 3 weeks, and of course in lecture in the meantime. Bye-bye. So Stuti and Andrew, are you still there? Yeah, perfect. So I'm going to create a breakout room just for the 3 of us so we can chat briefly. And then Tatiana, actually, you know what, Tatiana, I'll just put you on the waiting room for a second. Is that all right?
Yeah.
And Evelyn? Okay, perfect. Okay, let me stop the recording.